Today I'm going to be going over the key features of the Watson and Crick DNA structure. This graphic provides a general overview of the structure of DNA, the right-handed double helix. DNA is composed of nucleotides, which I will reference later in the presentation. The sugar phosphate backbone is appropriately composed of deoxyribose sugars and phosphate groups. The nitrogenous bases that make up the central part of DNA are adenine, thymine, guanine, and cytosine. In the DNA graphic, you will see that there are specific pairings between these bases that I will now go over. In DNA, the base adenine will always pair with thymine and guanine will always pair with cytosine to form the central part of DNA. This image shows the structures of all the nitrogenous bases. Each pairs between a purine and a pyrimidine. The purines are adenine and guanine and are recognized by their 6-member ring connected to a 5-member ring. The pyrimidines are thymine and cytosine and have one 6-member ring. These pairs of nitrogenous bases hydrogen bond with each other, which contributes to the stability of the DNA molecule. The hydrogen bonds between the bases is indicated by the dashed lines in the images. Now this is a section of DNA showing off all of the components. First, there's the phosphate groups that make up the backbone of DNA. Then there's the deoxyribose sugars that are shown in blue that connect to these phosphate groups and the bases. In this image, it shows the nucleotides for DNA and RNA. DNA has deoxyribose sugars, where it is missing a hydroxyl group on the 2' carbon that the RNA ribose sugar has. The 3' carbon on the deoxyribose sugar connects to the other nucleotides at the 5' end and forms the sugar phosphate backbone. As the backbone links together, the planar base pairs stick closely on top of each other due to hydrophobic interactions, which stabilizes the structure. In the center, we have the nitrogenous bases. Due to the anti-parallel arrangement of DNA, the structure has major and minor grooves, which are important areas for binding by proteins to DNA. The DNA double helix is a right-handed helix. To determine if a double helix spiral is right-handed or left-handed, you can use the right-hand rule. By putting your thumb in the direction of the spiral using either the left hand or the right hand, you can curl your fingers in the direction of the spiral, and if it matches, then you know whether it's left-handed helix or right-handed helix, depending on the hand used. In this example, we see that this is a left-handed helix, showing off my left-handed rule. And we have two more examples of left-handed spirals matching the left-handed rule. The fourth example also matching the left-handed rule. And finally, the last spiral staircase showing off that it is a left-handed helix.